Understanding the human immune system and combating diseases like cancer can be complex. Before we start, we would like to introduce you to the key characters in this video. We'll talk a lot about interleukin-2, or IL-2 for short. IL-2 is part of a large family of signaling proteins called cytokines. IL-2 contains 3 domains that dock on the receptor subunits of immune cells. 2 of these domains bind to CD132 and CD122 receptor subunits, telling immune cells to attack disease. The third domain binds to the CD25 receptor subunit, telling different immune cells to slow down an immune response. Basically, IL-2 signals to cells what to do. Originally, IL-2 was identified as T-cell growth factor because its primary function is to signal T cells to grow. Regulatory T cells, or Tregs, are a specialized subpopulation of T cells that act to slow down an immune response. Simply put, Tregs help prevent our body's immune system from attacking itself. Scientists also discovered that IL-2 plays a significant role in the immune system and in fighting disease. After immune activation, IL-2 binds its receptors on the surface of effector T cells and natural killer cells to promote a stronger biological response. Effector T cells and NK cells are vital players in mounting an immune response. They help our body's immune system fight diseases such as cancer. There's a key point for you to remember in this video. A single Treg can suppress many effector cells. For example, if one Treg can suppress up to 10 effector cells, then 2 Tregs can suppress up to 20 effector cells, and so goes the cycle. Pretty soon, this suppression can limit the body's immune system to fight diseases. That's the basic science behind how our body's cellular immune system works. In the fight against cancer, what if artificial intelligence could help design a drug to meet one of the biggest challenges in immunotherapy? Safely harnessing the power of IL-2. Interleukin-2 is often described as a double-edged sword because of its ability to both suppress and activate the immune system. Scientists and doctors have known for decades that recombinant human IL-2 therapy has tremendous power and limitations. When given at very high doses, IL-2 therapy has demonstrated improvement in response rates and survival in patients living with certain cancers. However, giving high doses of IL-2 comes at a cost. Outside the tumor microenvironment, large amounts of IL-2 often cause serious side effects, including vascular leak syndrome, low blood pressure, and swelling in the lungs and throughout the body. Let's see what's happening in the tumor microenvironment. As IL-2 appears in the tumor microenvironment, most of it heads toward regulatory T cells. That's because Tregs have a higher affinity, or stickiness, for IL-2 than effector T cells and natural killer cells. This results in Tregs consuming more IL-2 than effector T cells and NK cells. As the Tregs consume IL-2, they multiply in the tumor microenvironment and the lymph nodes around it, outbalancing and suppressing the effector T cells and NK cells, leaving many of the cancer-fighting cells unable to attack the tumor. Now, some of the IL-2 does make its way to the effector T cells, which stimulates them to produce more IL-2. But here's the catch. Most of this new IL-2 is consumed by the Tregs, keeping the immune system suppressed. At Allos, we call this the negative feedback loop. What if we can break the negative feedback loop? Based on clinical data, our human monoclonal antibody, imnescabart, could be the answer. Engineering antibodies with the exquisite precision needed to control IL-2 was beyond the capabilities of existing technologies. With the assistance of artificial intelligence and machine learning algorithms, imnescabart has been designed to attach to the part of IL-2 that binds to the CD25 receptor subunit. This prevents IL-2 from binding to Tregs, vasculature, and eosinophils, while still allowing IL-2 to bind to effector T cells and NK cells. When imnescabart enters the tumor microenvironment, it binds to the CD25 domain on IL-2, preventing IL-2 from delivering a growth signal to Tregs. Instead, the IL-2 imnescabart complex can only bind to cancer-fighting effector T cells and NK cells. As the effector T cells are activated, they release more IL-2. Imnescabart binds to this newly produced IL-2. This prevents Tregs from consuming the IL-2 and instead be consumed by effector T cells and NK cells. The balance has now shifted toward the cancer-fighting effector T cells and NK cells, creating a positive feedback loop that fights cancer. Outside of the tumor microenvironment, imnescabart prevents IL-2 from binding to vasculature and eosinophils. This is designed to reduce the severe side effects associated with IL-2. Imnescabart is the first human monoclonal antibody designed with the assistance of artificial intelligence to enter a clinical trial. Clinical data showed that imnescabart can safely harness the immune system to activate effector T cells and NK cells to fight cancer, reduce Tregs, transform the negative feedback loop into a positive feedback loop, and limit vascular leakage. These findings present a completely unique profile in the IL-2 class. At Allos, we're setting a new direction for IL-2 immuno-oncology so patients can live longer and better lives.